Something is deeply wrong with the rules of life at the bottom of the ocean. Surface worms are barely finger-length, yet 2,500 meters down, scientists found giants as tall as men, where no skeleton or known biology should allow it. The deeper you go, the bigger these creatures get, defying everything we know about animal limits. Why do worms break the laws of size under crushing pressure and freezing darkness? And what secret force makes the impossible possible here? The answer begins with a single, shattering discovery on the ocean floor. In 1977, the deep-sea submersible Alvin descended to the Galapagos Rift, 2,500 meters below the surface. What the team found there was so out of scale, it forced a rethink of what animals could become. Fields of giant tube worms, Riftia pachyptila, stood upright in dense clusters, some stretching nearly 3 meters, taller than most of the scientists who discovered them. Until then, the largest marine worms known from shallower waters rarely reached more than 10 centimeters. The contrast was immediate and unmistakable. In the abyss, worms were not just a little bigger, they were dozens of times larger. The discovery felt impossible. What made the finding stranger was the absence of intermediate giants at shallower depths. In the ocean's upper 1,000 meters, polycate worms and their kin remained modest, rarely exceeding a few centimeters in length. At hydrothermal vents far below, Riftia and its relatives reached lengths that seemed impossible for soft-bodied animals with no skeleton. The difference was not subtle. It was a leap, a boundary that only the deep seemed to cross. That stark boundary pointed to a dramatic change in conditions, not a slow gradient. This pattern was not limited to a single site or a single species. Across hydrothermal vent fields from the East Pacific Rise to the Galapagos Rift, wherever the depth exceeded 2,000 meters, these immense worms appeared. Their size was not a fluke of one population, but a consistent feature of life at these depths. The deeper the vent, the more likely it was to host worms of astonishing proportions, while the shallow ocean remained a world of miniatures. The search for an explanation therefore shifted. Scientists stopped looking for slight anatomical tweaks and started looking for something unique about the deepest, coldest, most chemically charged parts of the sea. What follows is the story of how that search led from mystery to mechanism, and how the ocean solved a problem that, at first glance, seemed to break every rule of biology. Cold water at depth changes the rules for life in ways that are not obvious from the surface. At temperatures just above freezing, between 2 and 4 degrees Celsius, seawater can hold far more dissolved oxygen than the warm sunlit waters of the tropics. The physics is simple. As water cools, its ability to carry gases increases. While tropical surface waters at 25 degrees Celsius rarely exceed 5 milligrams of oxygen per liter, deep ocean water at vent fields can reach around 7 milligrams per liter, a 40% jump. This difference matters. For animals like tube worms, oxygen is the key ingredient for powering any kind of metabolism, and at the surface, its scarcity places a hard cap on how large a worm can grow. In the abyss, that ceiling lifts. The deep ocean's cold, oxygen-rich water removes one of the classic constraints on animal size. Here, worms can extract more oxygen with every breath, fueling tissues that would suffocate in warmer, less saturated water. This advantage is not just theoretical. Oceanographers measure oxygen levels at these depths with sensors lowered from research ships, and the results are consistent across vent sites from the East Pacific Rise to the Galapagos Rift. The cold itself slows metabolism, but the abundance of oxygen means that, for the first time, Size is limited not by what the animal can breathe, but by what it can eat. With the oxygen barrier lowered, the puzzle narrows. The question is no longer how giant worms survive in the deep, but how they find enough energy to build such massive bodies in a place where food, in the traditional sense, is almost non-existent. Dissection of a giant tube worm reveals a body plan that confounds expectation. There is no mouth, no stomach, and no trace of a digestive tract, features that define almost every other animal. Instead, the interior is dominated by a single, sprawling organ, the trophosome. This organ fills nearly half the worm's body volume, stretching from just below the bright red plume all the way down the trunk. Packed within the spongy tissue of the trophosome are billions of bacteria, so densely concentrated that under a microscope, the host's own cells are almost lost among them. For decades, biologists struggled to make sense of this arrangement. Without a mouth or gut, there is no way for the worm to ingest food, no path for nutrients to reach its tissues by ordinary means. The absence of an anus means nothing leaves the body through digestion either. Yet these animals are not only alive, they are among the largest invertebrates ever recorded. The scale of the bacterial population inside each worm is staggering. Estimates suggest that a single adult riftia can host more than 1 billion bacterial cells per gram of trophosome, with the entire organ accounting for nearly half the animal's mass. Every cubic centimeter of trophosome is a dense, living matrix of symbionts and host tissue, a biological factory operating in total darkness, cut off from the food webs that sustain most life in the ocean. This anatomical paradox raised a question that would drive research for years. If these worms cannot eat, and if their bodies are given over to a mass of bacteria, where do they get the energy to grow so large? The answer would come not from the structure of the worm itself, but from the chemistry of the world it inhabits. In 1981, a team of researchers finally cracked the code behind the giant tube worm's impossible growth. The answer lay inside the trophosome, where billions of bacteria were found to be running a process called chemosynthesis. These bacteria use hydrogen sulfide, abundant in vent fluids but toxic to most life, as their fuel. By oxidizing hydrogen sulfide with oxygen, the bacteria generate energy and fix carbon dioxide into sugars and other organic compounds. This process, once thought to be exclusive to certain microbes, was now powering the largest invertebrates in the deep sea. The worms themselves act as living reactors. Their bright red plumes are packed with a specialized hemoglobin, unlike any found in surface animals. This hemoglobin can bind both oxygen and hydrogen sulfide at the same time, ferrying both gases through the worm's circulatory system without being poisoned. The dual binding ability is essential. Oxygen is needed for the bacteria to oxidize sulfide, while hydrogen sulfide is the energy source. The hemoglobin's structure prevents the usual chemical sabotage that sulfide inflicts on oxygen-carrying proteins. Once inside the trophosome, the bacteria convert these raw materials into organic molecules which the worm absorbs directly. There is no eating, no digestion, just a direct transfer of energy from chemical reactions to worm tissue. The cold temperatures at vent depths slow the worm's metabolism to about 20% of what a similar-sized animal would require at the surface. This means the energy harvested from sulfide can be invested almost entirely in growth, not in keeping up with the high metabolic demands of warmer, more active environments. With an endless supply of chemical energy and minimal costs, the worms can reach sizes that would be impossible anywhere else. At 2,500 meters below the surface, The pressure is more than 250 times what we experience on land. That is enough to collapse most familiar life forms in an instant. Yet giant tube worms thrive here, their bodies built almost entirely from water-rich tissue. With no internal air spaces, they are immune to the crushing force that would flatten a gas-filled animal. Their cells are stabilized by specialized molecules known as piezolytes, which keep proteins and membranes functioning even as pressure mounts. This gel-like structure, nearly 95% incompressible water, allows the worms to remain flexible and intact where most creatures cannot survive. The result is a body plan that turns the deep ocean's greatest physical barrier into just another feature of the landscape, clearing the way for extraordinary growth. Direct observation at deep-sea vent sites has shown that giant tube worms can reach their full size, over 2 meters long, in just a few years. Researchers tracking new vent eruptions found these animals appear rapidly and grow at rates that rival the fastest marine invertebrates known. While shallow water worms take years to gain a few centimeters, Riftia populations colonize fresh vents and reach enormous size in a fraction of that time. Comparative studies across deep-sea animals, like giant isopods and colossal squid, reveal a consistent pattern. Cold temperatures, high oxygen, and abundant chemical energy support larger bodies and longer lifespans than surface relatives. DNA evidence from both worms and their symbiotic bacteria points to a partnership that stretches back more than 100 million years, giving evolution ample time to refine this extraordinary system. Models of vent chemistry and energy flow confirm that the resources available at these depths are more than enough to sustain such giants. Today, deep-sea vents remind us that life rewrites its own rules, using chemistry and not sunlight to build giants in the darkness. As we probe further into Earth's extremes, each discovery proves that what seems impossible is often just unexplained. The deep ocean still holds secrets that could redefine what is possible anywhere. Share your thoughts below.